Good morning, everyone. Um, so this is lecture 4 out of 5, and I was telling you yesterday. Um, there was a major shift in my professional life after I started dealing with the experimental data that Siddhartha Ribeiro generously shared with us. So years passed by in which we couldn't make reasonable models to those data, and then understood that having data was something precious, and that some other labs have other agendas. It could be nice to have our own data. So Pedro Carelli was convinced to apply to a position in Recife. He came, he is an experimentalist. And then later on, Ivaldo Vasconcelos joined us, first as a postdoc coming back from Portugal and bringing new techniques. I'm going to mention that. And nowadays, he's a faculty as well. And so what I'm showing here is the result after this whole process. And then applying to grants, to buy equipment, then to set up a lab from scratch, from an empty room, and training students to do many things. Things go wrong and then wrong. Eventually, they turn out to be okay. So those results I showed you yesterday were from paper from 2010, and they started way early, like 2007 or so. Results I'm going to show today are from a paper that came out from our own, from data generated in our own lab in 2019. And meanwhile, we did other stuff, but sometimes things take time. And so that's why there are so many pictures here, because what I talked about yesterday was in the first lecture, second lecture, was theoretical work with Mkinoichi. And then once you have a lab, then many people got involved, and I'm going to mention some of those names. We have collaborators in Portugal from where Nivaldo did his postdoc in Nuno Souza and Ana João's lab. So it's a lot of work. Many people are involved. I just want to make sure that people understand there's a lot of work behind that. So when I first mentioned Bags and Plains' work in 2003, and when they observed those power laws with those very nice 1.5 exponents, then I mentioned that this kind of absorbing to active phase transition gives you those exponents. This was well known. And then I told the story of how the model worked nice, and then how it fails to address more realistic problems. And there are some other issues. So experimentally, even for reduced preparations, I mean, with the animal is not freely behaving, you have very strong deviations from these values predicted by that simple model that was in the back of the mind of many people. For instance, you take this paper by Woody Shu in 2015, in which it's a very peculiar, I think, experimental preparation. This is the ex vivo turtle visual cortex. So they remove the whole brain out of the turtle head, and they keep it alive. I don't know how this works. And then they take part of the visual cortex, put a multi-electrode array here, and then play a movie to the retina of the turtle and measure activity here. Okay? And then you have electrical activity, you have many channels, you do the thing that I mentioned before. I'm still talking about neuronal avalanches, and today's the last day I'm going to talk about neuronal avalanches. So we'll see that it has problems, and then tomorrow we'll do something else based on Bill's idea that the things that I explained yesterday. So, so you have these local field potentials, you threshold them, you have activity, you choose some bin, and you have avalanches for which you estimate the size of those avalanches, and then the duration. You do probability distribution of those two things. You try to fit probability distribution. If it's a power law, then the exponent governing the size distributions. If it's a power law, The exponent is minus tau, and for the duration distribution, the exponent, it's called minus tau t. So they measured it for a bunch of turtles, or turtle brains. And what do you get? Not only results can be far away from what is predicted by this standard model of this brain criticality, actually have a plane of exponents. This is how they presented their results, okay? This is how they presented them. And tau 2 equals 2 is here. Tau equals 1.5 is not even in the graph. You have to extend it. So this is what mean field right speculation predicts. And you have this bunch of exponents there. So it doesn't look good as a model to explain things. And besides, I also showed you yesterday, I hinted at it briefly, but remember the things I showed yesterday in which you have less synchronous activity, like in waking behavior and freely behavior, and more synchronous activity, like this ketamine-xylazine anesthesia. Right? So for these more synchronous results, you have beautiful power laws. For these less synchronous results, you don't have the, perhaps due to undersampling, perhaps not, I don't know, you get this log normals. So the signature of criticality, the traditional one, which is power law avalanche size distributions, they are not there, they're not here, but they seem to be more plausibly here. So it's strange, it's complicated. The exponents vary. And so what was going on? We didn't quite understand. And so probably this thing was under jeopardy, this idea, this theoretical idea. And that's fine. This is when Nivaldo Vasconcelos came back from Portugal, bringing with him this technique of recording with more channels, more finely. I'm going to comment on that. But since I've talked about these oscillations and this synchronization kind of thing, perhaps it's nice to review, for those not familiar with neuroscience, this notion of sometimes they're called brain waves or something. The idea that oscillations appear everywhere in the brain all the time. And there's a whole literature about it. So EEG has just turned 100 years old as an experimental technique. This is one channel. This is a reference channel of 10 hertz. This is measurement. I don't know where, but I will guess occipital cortex here, which is where 10 hertz used to appear. This is a modern version. I don't know if it's state of the art anymore, but you can record from several channels. What is the advantage? It's non-invasive. You can do it with babies. And what is the disadvantage? Well, the signal is ugly because it has to go through the skull, the skin, right? So that's life. Make your choices. I took this from some advertisement for Father's Day back in the day. You could buy one of these a few channels here to improve your meditation or something, some kind of biofeedback. Doesn't matter. What was nice was that in this commercial product page, you can find a nice summary of what these waves are interpreted to be. You can also find it on Wikipedia, of course, but I mean, so you have different frequencies in EEG, which in the last century, since EEG was invented, people came to associate these frequencies to some behavior. So alpha, be something, so the bands have limits which are quite arbitrarily chosen. So it's from 8 to 13 or something for EEG. And it's physically, mentally relaxed. So this is one of the basic experiments, tests of EEG. You put your electrodes, this helmet in your head, and you monitor for primary visual cortex, which is in the back of your head. If you close your eyes, an alpha peak will emerge in your EEG. So one of the first was, oh, you close your eyes, you relax, so there's an alpha peak. And gamma, which is high frequency, 3200 hertz, is connected to heightened perception, learning, problem solving, blah, blah, blah. Okay, so there's a whole plethora of results, and it depends on where you're measuring. It depends on whether it's EEG or local field potential. So delta in local field potentials in the hippocampus of the rats or the mouse are associated with something and something, right? So people accumulated this kind of connection between frequencies and behavior, or a behavior state like being awakened or being, where is it? Deep sleep here, for instance, right? Anyway, these things, I think, costed like $100 or so. This comes from a more respectable source, some kind of review article in which they show that you have the neurons here in different cortical layers. If you measure local field potential at the surface, this is the kind of signal you're going to see. If you have electrodes measuring something deeper here, this is the kind of local field potential you're going to measure. And this is what spike measured with an electrode inside the neuron and outside the neuron would look like, right? So you can see, appreciate the difference in amplitudes and the quality of the signs, the signals. This is where spikes. If you're, again here, this is EEG, local field potential, and spikes now measured extracellularly, like most of the things I'm showing and Bill referred to. So you put an electrode outside the neuron and you measure from the consequences of the spikes and the flux of currents that appear, right? So this, from here, the picture is not, I apologize for the resolution of the picture, but the picture is not very good here. This is spikes measured extracellularly. This is measured intracellularly. Of course, you love to see this, but you don't, how do? You cannot insert electrodes inside the neurons of many neurons. So at the same time and expect this. It's a very, very difficult measurement to make. And this is spikes once you have processed this signal here, like spike sorting and stuff like this. So Bill mentioned state-of-the-art techniques. I'm just going to repeat a little bit with history along the way. This is a very bad picture, taken in 2013 in our lab, in our then nascent lab. It was just starting. And this is a 5-cent coin for scale. And this is an electrode array in preparation. So the way we learned to do this was from techniques borrowed from the group in Minas Gerais and in Natal. So you buy some silica tubes, which I don't know, 50 micron diameter. You glue them in a 4x8 matrix, so you have a model. Then you buy tungsten wires and you put tungsten inside each of those tubes. So you have a 4x8 matrix of tungsten wire. You fix this thing with glue or something. Thais Feliciano, who's currently at the NIH in Dietmar Planck's group as a postdoc, was the one who could do this. And then you solder all the tungsten wires to an adapter, which will then be connected to the electrophysiology data signal processing apparatus. Okay, so this is mid-preparation. It's being glued here, it's waiting, and then once it's ready, then you cut this thing with the appropriate length, and in a more nice picture, it looks like this. And then you connect with, well, you have to remove this thing, and then there's an adapter, and you make a surgery, there's a whole thing about the implant of these electrodes to the rat brain, and then the rat recovers, and then it can do experiments. So, of course, you can buy this thing. Then it costs you, this one back in the day cost like $500, which for us is a lot. So if you buy it ready from the company itself, if you make it at home, it costs like 10 times as less, $35 to $50. Of course, you have to make them. And the signal-to-noise ratio is not very good, but that was what was available at the time for us. And this thing has the advantage of you can do experiments in freely behaving animals, for which I'm not going to show any results. So I'm not going to talk about these freely behaving animals. Right, so this is what you see in a computer lab when the animal is doing something. In order for this to work, this wire here, which comes out of the electrodes, have to go to a special machine which has this is our older setup which had only 32 channels, and you have to have some commutator here which allows the mouse to go like this, and the cable keeps rotating while keeping the 32-tone contacts intact. Because if you don't do this, the rat can just chew on the thing and destroy the whole apparatus. Okay. And anyway, this is how we started doing things many years ago. And then, by then, silicon probes were already coming out. So silicon probes, you can only buy these things. Here we have I'm sorry, I didn't mention it before, but the spacing between the electrodes before is like 150 or 200 microns across between the electrodes. Here you get the electrodes much closer, the channels much closer, and therefore you can make much denser recordings. So when this thing is measuring, and look, the characteristic size of a body cell is like 10 microns, right? 10 to 20 microns. So if they're standing here in the middle, you get, it's the order of magnitude of a cell body. So this is much denser, and you're hoping you're getting most of the spikes which are occurring around these channels. So this is what we used in the results I'm going to show you. Both data obtained in our lab, which is a technology that Nivaldo Vasconcelos brought from Portugal, and data from his former lab in Portugal. So we had data from different labs, different number of channels, and different strains of rats to compare. So that was nice. I mean, more recently, we have the NeuroPixels, which has the same idea, roughly, in the sense that you have these channels here, except that it has more channels, and you can choose on the software which ones are going to be active once you insert the electrode. So it's even cooler and more expensive. The things that you have to consider, and I always like to remind people of that. I'm going to show results of anesthetized rats, okay? In a minute. Sorry. With this kind of technology. And sometimes you talk to theoretical physicists or who are not familiar with these things. Say, oh, come on, why don't you do a freely behaving? It's much nicer. I don't like anesthetized animal. Yeah. If you want to do freely behaving, unless you do this technique now with the head fixed, which is a more recent technique, if you want to insert this electrode on the head of a awake animal, you have to fix the electrode on the animal head with cement. That's how people do. Dentistry cement. And then when the experiment is over, the animal eventually will be sacrificed, your electrode goes away. You don't recover that thing in this setup. It costs $1,000. Some labs can afford to do that. Some labs do. I can't, right? So just a reality check. Sometimes you have ideas, and then you have to go to reality. Right? Anyway, And, this thing, on the other hand, this is affordable, but then the signal-to-noise ratio is bad. So pick your choice, right? A more realistic experimental setup with a behaving animal doing all kinds of cool stuff, but poor signal, or better signal. And you can have both if you use this other technique with the head fixed. So I'm going to talk. Bill mentioned that yesterday, or the day before yesterday, and I'm going to come to that in a minute, tomorrow, I think. So for today, so let's go back to brain waves for a while and spikes. So there's this notion of cortical state, which is nicely reviewed in this paper here. I like it very much. So let's read it together. The brain continuously adapts its processing machinery to behavioral demands. To achieve this, it rapidly modulates the operating mode of cortical circuits, controlling the way the information is transformed and routed. Well, so the notion of state here is not quite the state we are used to in physics, like the state in Hilbert space or a dot in phase space X and P, right? For Hamiltonian dynamics or something like this. It's more akin to a thermodynamic state in which it's liquid or is it gas. So a state here is the dynamics of network activity on a time scale of seconds, is how the network is behaving, what kind of behavior the network is presenting if you look at it for a couple of seconds. Okay, this is what they call a state in this literature. So if you were to simplify a lot, they have this called synchronized state, which they're displaying here, which is what you observe when the animal is drowsy, quiescent, or during slow wave sleep. As opposed to desynchronized state, which is what you observe when the animal is actively behaving, alert. I'm mentioning essentially rats and mice, which are not the same. So how do people characterize that? I mean, you can look right away. For instance, this. If you look at local field potentials, you see these low frequency fluctuations. Whereas here you see less low frequency local field potential fluctuations. And there are ways to quantify that. Okay? If you look at spikes, you can see that in order for this local field potential, I mean associated to local field potential, like low frequency fluctuations, you see this correlated spike in activity, right? I mean, they're not all together, but they're kind of correlated. And you can measure that. So have N neurons, like Bill was mentioning yesterday, in order to do maximal entropy, right? So if you have N neurons, have N minus 1 over 2 pairs. You can calculate the correlation between those pairs. And in this case, they make a histogram of this pairwise correlation. And you see that it's essentially positive correlations, mostly. Not everybody have a tail here, but you tend to have a shift to positive correlations because when they don't spike, they don't spike. When they spike, more or less altogether, more or less, right? Whereas here, if you take this histogram of pairwise correlation, what you will observe is something with mean around 0, close to 0. Not necessarily strictly 0, but you have positive correlations and negative correlations and a little bit of everything. And this characteristic nearly steady and fluctuating local field potential and average firing rate, population firing rate as well, like I showed you yesterday for those free behaving animals data that I got from Siddhartha Ribeiro. Okay? Now, as it turns out, when I'm quoting from this paper, cortical state not bimodal. There's no such a thing as this or that. It's more like the separation is kind of a bit arbitrary. There's some kind of continuum there. understood to some extent. And we asked ourselves whether we could explore this continuum to try to solve this conundrum between criticality and synchronization that seemed to appear in that previous data. But that previous data had this issue of undersampling. That was old data with older technology. Maybe with new electrodes, with silicon probes, denser recordings, more spikes, more neurons, maybe we could have a look at this thing again. And if so, how can we do this with some under control, since we cannot do the experiments with free behaving animals for the reasons that I explained before? Can I do it somewhere, some other way? And so this is, again, input from, insight from Nivaldo Vasconcelos. You could use urethane. So urethane is a particular kind of anesthetic, which was discovered in explored more deeply in the early 2000s, in which neuroscientists regard as some kind of, if you anesthetize a rat with urethane, it becomes what they call an animal model for the translation between like slow wave sleep and REM sleep, or between slow wave sleep and wakefulness, between things which are more synchronized and things which are less synchronized. Because under urethane, the animal undergoes these transitions spontaneously. Let me show you the plot. So, results I'm going to show you are from rats which have been anesthetized with urethane, and they have been implanted in the primary visual cortex here with either 32 or 64 electrode silicon probes, and then you measure brain activity. So the first plot I'm going to show you is not from brain activity itself, but rather from something else, from the coefficient of variation. Coefficient of variation of anything is just the standard deviation divided by the mean, so it's a dimensionless number. Of what? I'm showing here the coefficient variation of the population firing rate, this data. So this is experimental data from the setup. Each line here corresponds to a neuron. Each different color corresponds to a different shank of this 4-shank or 6-shank, I don't know, electrodes. And this is time scale, like 1 second here. And the line is the sum activity of all neurons at a given time, a given instant. So this is called instantaneous firing rate, population firing rate. Okay? So this line here, I agree, Bill, with you that the colors have changed since the first days, right? The colors have changed. No, yeah. So this purple thing corresponds to this purple point here, which has a low coefficient of variation. Why? Because if you look at this plot, you have some fluctuations of the instantaneous firing rate, but the variance here is small. The standard deviation is small as compared to the mean. If you divide the standard deviation by the mean, you get some low number. Okay? At the other extreme, take this green guy here, which has a high coefficient of variation, like 2 or something. The other one has 0.5 or something like this. This one has like 2. And this guy is high synchronization. And so when they're silent for everybody. When they go up more or less together. So this thing fluctuates a lot. Therefore, if you calculate the standard deviation of the population time series, it's very high, divided by the mean. So this is high CV. This is low CV. I'll be talking about CV all the time. So CV is that, it's coefficient of variation of the population firing rate. And you have everything in between. And this is an intermediate CV like this. This is low CV, this is high CV, this is intermediate CV, and you have everything in between. This is I think you will agree with me, this is less synchronous and more synchronous. Actually, if you write down the equation for the standard deviation of the population fire rate, you see that it depends on the pairwise correlations and whatnot. So it's consistent, okay? It makes sense. It's just that it's easier to show one number instead of the whole probability distribution of the pairwise correlations. But I didn't bring the plot, but so we do calculate the pairwise correlations between all the pairs and show that if as you go from here to here, it is a near zero histogram to near a zero mean histogram to something positive here. So as you can see, this is the time series for coefficient variation of one animal for about 3 hours or so. This is seconds. And it changes in an unpredictable way. And you take 10 different animals, they have 10 different curves like this, more or less unpredictably. But what you do know is that they don't stay fixed on a single quote unquote cortical state, like asynchronous cortical state like this, or synchronous cortical state like this. They go around, they wander around, and we thought we could explore these possibilities to understand about what's going on. So, what we're going to do is we're going to parse this data according to CV values, finer detail than I'm showing here, and do whatever we have to do. So the first result I'm going to show is about avalanche dynamics, because that's what people were doing all the time, except we're going with more to include 10 years later or many years later, more sophisticated analysis of these avalanche results. So you have this spike avalanche, so we're going to do the same thing again, define an appropriate mean, calculate the size and duration of the size and lifetime of the avalanche, and calculate distributions. And depending on the value of CV, here I'm showing 3 values, but we have these more fine-grained slices. You can have different distributions. Some look like reasonable power laws, some others don't, like low CV typically don't, like we had seen before, even with denser recordings. But so you do those feeds, you make a statistical test to see whether it's the best fit or not, log likelihood estimator and whatnot, blah, 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 right? So when you do this, you see that you get some exponents out of it. This is what's predicted by mean field error percolation. This is what getting the data. Perhaps this one is compatible, perhaps this one is not. What matters is that as you change the CV value that you're looking at, these exponents change more or less continuously. And this reminds you of those third results on one hand, and on the other hand, raises a red flag from your statistical mechanics course. Okay. And if you have an universality class, you have one exponent, right? Unless you have Griffith's phasor. Okay, yeah. So is there a criterion to choose which are the true exponents? So by then, people were going back to physics and going beyond just looking at one exponent and drawing conclusions from this. So essentially, so assume that you have found some decent power laws, just assume, and that therefore you have this power law distributions for size and for lifetime. Then the average size given a lifetime should also scale like this. This is a combination of exponents. Okay, I didn't call it gamma. People call it gamma because gamma is something else. Gamma is for stability. I don't find out alphabets. Anyway, so this should scale with, this is reasonable, right? The longer the avalanche, the larger its size. Except that at a critical point, this dependence is nonlinear in principle. And we also obtain that. So the average size for a given duration, it has some non-trivial slope here, the log-log plot, and this exponent here also depends on CV. Okay? Now, more important perhaps than this power law behavior, this power law behavior, and this power law behavior is the fact that if the system is really critical, according to theory, you can do the math, these exponents are not independent, but they are connected through a scaling relation. So there is a scaling relation between them. So this is old news in statistical physics, but it was news in 2012 when John Baggs and collaborators just tested this with some data from their lab. So by then there were many people protesting this criticality, thinking, oh yeah, you can get this power law in many different ways. You can get this power law in many different ways. Perhaps this one. Yeah, so, yeah. But then this one is harder to get. Okay? Get one power law, another power law, and the three power laws, it's okay, and then get the exponents of the three to agree. It's something else. So this was considered a higher bar for the data. Okay. Okay? So, note that experimentally, for us, the two sides of the equation, the scaling relation, this is called the correctly noise scaling relation. Okay? This guy depends on CV, this guy depends on CV, and this guy depends on CV, and they come from different measurements. So, this guy I obtain from plots like this guy I obtain from plots like this, and this guy I obtain from plots like this, and they in principle are independent. And so, the brain activity of this animal is moving around, being more synchronous, less synchronous, more synchronous, less synchronous, I don't know, more or less continuously, and I'm measuring these exponents as they change. And I collect them as a function of how synchronous or less synchronous it is as measured by coefficient of variation, by CV. Okay? And this thing is changing, this thing is changing. Is scalar relation satisfied as CV changes? So this guy depends on CV. I'm going to make a plot of this guy as a function of CV. Left-hand side of the equation as a function of V. I'm going to make another plot of the right-hand side of this equation as measured experimentally as a function of CV. And the question is whether they cross. Okay? And we were very happy to see that, yes, they do. They do here. So different symbols here correspond to different animals. And this is like the group data if you take this kind of average. So in this region here, so you have, okay, so the fact that you have curves here means that you have all kinds of exponents just measured, subjected to statistical tests, but still just measured blindly without forcing an exponent value mean field error calculation. I don't care, I just want to see what comes out. And here you get this region, this gray region, in which the two exponents, the two sides of the equation agree as some kind of intermediate CV value. And what is, so this satisfies this scaling relation, which is expected at a critical point. So this was considered as a signature of a more respectable signature, perhaps, of a critical point, in the sense that it satisfied a higher bar. It passed a higher bar, theoretical bar. Okay? And what is more interesting is that the residence time, if you collect all the rats and stuff, peaks precisely where this agreement occurs. The interpretation, of course, gets complicated because if I say that the system is critical here, then it's critical here in passing, en passant. And then it goes away, and then it comes here, and then it goes away, and right? Notice that as I say these words, I'm more or less implicitly understanding CV as a sort of control parameter that I do not control. And this, spoiler, I'm going to convince, well, we had to convince ourselves, and then I'm going to convince yourself, this is actually not the case. So for now, let's keep this image in mind, and then I'll revise it. Of course, there are always third referees, which ask you to do more. I don't like anesthesia, blah, blah, blah. So I got an anesthetized macaque monkey from somewhere in the internet, which was freely available, and got the same thing here. And freely moving mice, again, data freely available, which show the same behavior, except that here you see the residence time is away from this supposedly critical point. Okay? So this is nice. Now, if we have a critical point here, what are the exponents? And are they compatible with this mean field direct calculation? So we had this question in mind about the universality class of this putative phase transition because people talked a lot about it. Nowadays, I'm not sure as I'm going to show you tomorrow, I'm not sure I care that much, actually, as much. But for now, we had this question, right? So I have many exponents here. So the exponents tau t and tau are combined in these white plots. So they have different values. But now I know which ones to, quote unquote, choose that satisfy the scaling relation. So what I'm showing you here is, again, like in Woody Schu's paper for the turtle, a plane of exponents tau and tau t to see how they spread. So please look at the blue stars first, which is our results. They appear to spread on a line. And when I saw this, I remembered Woody Schu's papers. I asked Antonio Fontanelli, who's the leading author of the paper, please grab those results from Schu lab and put it on the same plot. And the ex vivo turtle is here, is more or less aligned. And the freely moving mice are here. The cross model that I briefly mentioned yesterday, I think, on the blackboard, with a more sophisticated model with long-range time correlations, gives an exponent here as well. And this is a mean field directed percolation. Okay? So for the anesthetized rats, this gray region here, where the scaling relation is satisfied, amounts to this blue region here. So tau is 1.5, which precisely mean field directed percolation. But tau t is not. And you can have the arrow bars. You see the arrow bars are here, right? So it's not compatible with mean field direct percolation. For the free moving mice, I should make a comment about this data. So this is not our data. We got it from the internet. And we don't know what these mice are doing. We don't know whether they are sleeping or not. We don't know what's going on. So many things could be going on with these mice, right? That we don't know, essentially. So there's little control there. These are different exponents. Here's borderline compatible with mean field direct percolation, this exponent, but this other one isn't. Here it's the other way around. So we were very happy with these results because we understood them to be some kind of signal to criticality, which had passed a higher bar, as I mentioned, because it satisfied the scaling relation, which is harder to do. And since results were incompatible with mean field direct speculation, said, well, okay, look, it is not. So new models to think about were required, and perhaps the cross model with this emergence of oscillations, like I mentioned yesterday very briefly, together with self-sustained activity, will be a candidate. What kind of explanation is this? I'm not sure. So there was work to be done. Besides, you have these different exponents for anesthetizing free, different experimental conditions, which is perhaps not surprising. And even away from criticality, these two exponents follow the same linear relations for different setups. We don't quite understand this spread of exponents along this line. This slope here actually coincides with 1 over sigma nu z, this third exponent. And like Miguel Munoz told me once, either this is really important or really bullshit or trivial. I don't know which one it is. I'm fine with both, actually. But this is the result we got just by applying the best fitting techniques for this paradigm or looking at avalanches. As I mentioned, I've been telling you repeatedly, has again and again methodological issues, right? That we thought we had circumvented by using denser recordings and more sophisticated theoretical tools like asking the data to satisfy scaling relation. So 3 power laws plus 1 scaling relation. Okay? We're very happy. One of my students was not so convinced. Tawan, he was just skeptic. And a very brilliant guy, very hardworking. So he didn't quite buy our story. So he decided to check it. What did he do? He took a clear mean field error calculation model. I'm going to show this model because this model is much more sophisticated than the one I showed you in the second lecture. Because this one has excitation and inhibition, right? It's more easy to talk to a biologist and convince him than it looks. It looks like a little bit like the Brunel model. Some people might know the Brunel model, but it's even, I think it's even more, has a nicer touch in that it has stochasticity in it. So he took, what I'm going to show you in the next slides is the idea that Tawan then tells in this paper. He took an MFDP model and subjected it to the same constraints experimental data. So he runs a huge simulation with lots of neurons, but he subjects it to severe subsampling, like the data always is subsampled. It is always undersampled. You're measuring hundreds of, so in this setup with these silicon probes, we had access to hundreds of neurons. I don't know how many hundreds, 100 to 150 neurons. Well, the rat has many more, right? Even only the visual cortex of the rat has many more. Even though that cortical columns where these electrodes are have many more. So he simulated large n, 10 to the 5 or something, and sampled small n, like 100 or so, the order of magnitude that the data we had. And then he just blindly subjected the data to the same thing that we do with the analysis pipeline. So parse by coefficient of variation. Well, first calculate coefficient of variation, parse by coefficient of variation, bin the data, fit power loss, check the exponent and its correlation. Like you didn't know the model belongs to the mean field error calculation universality class, and therefore you know all the exponents from start, from day 0. In principle, you don't have to do anything. You know the exponents analytically. And I'll show the exponents, how beautiful they are if you take the whole model. So this is the model that he used. It was a model proposed by, again, Osami Kenouchi, Mauricio Jardim and collaborators here. It's essentially a leaky model for the membrane potential here. This is mu is less than 1. Then this VI is decaying exponentially, plus some input. And then you have excitatory connection from excitatory neurons and inhibitory connection from inhibitory neurons. And Pretty much like in the Brunel model, you control the relative intensity of these two, the excitation inhibition via this parameter G. X is a binary variable. It's spiking or it's not spiking at time t. It's a discrete time model. And the difference to the Brunel model is the fact that once you have this membrane potential for a given neuron, the probability that it fires is not a step like this, but it has a graded sigmoid-like function. So here it's probabilistic, and this makes the model. It can have a clear second order phase transition, which they calculate, which in the all-to-all coupling they calculate analytically. It's very nice work, theoretical work, very simple, but you know. So here I have a plot of the second order phase transition, the mean density of active sites as a function of this inhibitory coupling here. And it's the other way around. I usually show plots like this. This is the other way around because it's inhibition. So we have the supercritical phase for low enough inhibition, a critical point here, which for, so you have lots of parameters that I'm not showing here, okay? But essentially, so you have, I don't know, 75% excitatory neurons, 25% inhibitory neurons, like in the cortex, or 80 to 20, I don't know. And you fix these other parameters, and you run, and you can calculate. So the critical value is exactly 1.5 of the coupling parameter inhibition. And here you have what we call a subcritical region. There's too much inhibition, it goes to an absorbing state. Okay? So if you take the model on an all-to-all coupling network with 10 to the, I don't know, 4 or 5 neurons, and you sample all the neurons, like we do usually in models, and you calculate, this is the kind of, well, instantaneous activity that you'll see. So take this subcritical point, too much inhibition. It's the purple line, so it goes to 0. Once it goes to 0, you flip one neuron at random, it goes up again, and then it goes to 0 again. And then you flip one neuron again, it goes to 0 again. So you keep flipping so that you keep the network alive, okay? So this is the limit of 0 external field in the jargon. It keeps fluctuating around here, low values. For the supercritical case, on the other hand, you have this self-sustained activity. Eventually, you go to 0 by chance, then you flip it again, ta, ta, tra, 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 tra. And in the critical case, the red curves, you have this thing in between. So if you want, each of these red peaks is kind of an avalanche, and you can do the statistics. You do statistics for everybody here. If you do the statistics for this model, this is the size distribution. You have this, as predicted by theory, 1.5 exponent over, I don't know, for many decades, 4 decades. Because you have a very large system. You can models are beautiful, right? You can run things, and theory is beautiful as well, because then you know what's going to happen, and it happens. And the time distribution is always, the lifetime distribution is always uglier than this one. But again, you have a nice power law with exponent 2, and if we do some advanced mathematics, so this is the scaling relation for the third exponent, right? 1 over sigma mu z. If this is satisfied, oh, if this is 2 and this is 1/2, this is 2. Okay? And that's what you get here, 2. This is just to convince you that if, despite the complications, well, not complications, but despite the fact that the model has excitation, inhibition, it has some leaking term, etc., it has only one absorbing state. And then there's a conjecture in this literature that all systems with a single absorbing state and no further symmetries should belong to the direct population universality class. This is not always the case, but it's mostly the case. So this one is more complicated, but it gives you the same universality class, and these plots are here to show you that. So we know the exponents. But what Tawan Carvalho wanted to convince me, after all, was that this guy, which is clearly mean field derived percolation, when subject to the same conditions that the experiments that we had for the rats, would fool us into believing that we were in a different universality class. That's the result. So these are the results for full sampling. Everything okay with theory. But now, what happens, you close your eyes, you forget, you pretend you don't know. These are the correct exponents. And you then subsample, undersample. I'm going to look at only like 100 neurons, which is more or less what we have experimentally, out of these 10 to the 5. And we do everything blindly, right? What do you get? Well, here's, in color, I'll show the results for the model, and here I'll show in a minute the data that I showed before, but I'm going to show again. So as you change this coupling parameter, so 1.5, 1.585, 1.7, as you decrease inhibition, the CV goes down a little bit. It fluctuates. CV calculated exactly in the same way as experimental data. So we take chunks of 10 seconds to calculate this CV for the population frame rate. And the first thing to notice here is that even, okay, this is the model parameter. We control the model completely, right? So parameter is fixed. But CV fluctuates. Even for fixed G equals 1.5, look at the distribution of CV that I get here. The thing that in the previous experimental results, I was considering a control parameter. But this is a true control parameter. This is a very crude proxy of that because it has a very wide distribution. I changed the control parameter here, and this distribution of CV values has a huge overlap with the previous one. And I come here and notice that CV here is varying from 0.5 something to 2.5, which is the range of experimental values that I had before. So I get this whole range of CV values just by changing my coupling values within 3% of the critical point. And now, if I only look at data, again, the one repeated the analysis pipeline that we had for the data, you just take, whenever your data collectively has CV something low or something like this, you do, let me do the raster plot and show you the population activity. This looks very asynchronous behavior. Low CV, exactly like we saw in urethane data. If I select windows in which CV turns out to be high, and I look at the data, this is what I get. I get this apparent oscillations. Remember, it's a mean-field direct percolation model. There are no oscillations in the model. Perhaps here the explanation you had the other day makes more sense, that you're looking at some 100 neurons, then activity comes to these 100 neurons, it goes somewhere else, and then comes back eventually. So you see these as then everybody going together, then going silent. They're not going silent. Activity is elsewhere where you're not looking. But it's, you see, so this model has no oscillations. But that's what you more or less see here, this collective synchronized activity. And then when you take this subsampled activity and do your avalanche stuff, you can get all kinds of stuff for low CV, higher CV, intermediate CV. You fit, you get some exponent, which is not 1.5. It's something that comes out of fitting. Here is 1.84. Here's 1.31, not 2, and not 1.5. You have all kinds of stuff. This is the experimental data. This is low CV, which looks like this. This is high CV, which more or less looks like this. These are the power laws with funny exponents and whatnot. The only thing that the model, as such, cannot reproduce is the fact that the time evolution of CV here is way richer than it is here. So one thing that we tried to do, but we didn't try perhaps enough or for long enough, was to put some dynamics in the coupling here, so it will jiggle around this point to see whether this will cause these different curves here to morph into something like this. We haven't tried it yet, but it's up for grabs if someone wants to do it. I don't know if it matters, but I know, right? But please notice that this interval of CV values generated by tiny changes in this actual coupling of the model, this whole range of CV covers pretty much experimental values, experimental range of values, right? And then once you have these exponents, which depend on CV of the subsampled model, you can do everything again and see whether this curve here, 1 of sigma mu z, is a function of CV, and this right-hand side is a function of CV, if they cross somewhere. This is the data. Again, okay, I'm sorry. The reason why the experimental results are different here from the one I showed before is that because here we are using data only produced in our lab. We finally upgraded our system from 32 to 64 electrodes. We bought new probes, and we wanted to test whether our data was as good as before, and it was. So this is like, it mattered to us, okay? But it's the same kind of results. Again, a spread of exponents in the plane, mean-field error calculation, the exponents, the crossing between the two sides of the scaling relation here, experimentally. And this is the model, subsample model as done by Taiwan. You see that you have all kinds of crazy exponents, all wrong. Okay, is that clear? Let me go back just to make this clear. This exponent is wrong. This one is wrong, and this one is wrong. It's distorted due to subsampling and whatever you're doing. The exponents are 1.5, 2, and 2 analytically, I showed before. So this is wrong. Yet, the 3 exponents are wrong. Yet, they combine in a very cruel way to satisfy a scaling relation, and therefore fool us into believing that we have non-trivial exponents because they satisfy a scaling relation. So apparently, that high bar was not high enough. Same what?
The top and bottom figure of the right-hand side, is that black dashed line the same slope? Yes or no?
The black dashed line is a fit.
It's a fit, okay, so it's not, because it's 1.3,
in the other case, it was 1.28.
Okay.
Yeah, it's the same, yeah.
I mean, it should, depend continuously on the level of subsampling. So the fact that all of your data from the monkeys and mice and things had the same slope means that somehow they're subsampled to the same extent,
so,
roughly.
Yeah. I think I'm not showing this, but that's one of the plots in the paper. As you increase subsampling, you see this cloud of exponents eventually marching toward mean-field error calculation, as they should. Yeah, so we have done that. Yeah? So we have this nasty, I don't know if it's nasty or it's obvious or both, but satisfaction of the scaling relations for completely wrong exponents, right? Only if you're not too far away from criticality in the original model. So there's that. If you're here, you're at 3% of criticality, then your exponents go away from this line and start flying somewhere else. So this curious behavior in the sense that, yes, this crossing, okay, maybe some kind of signature of criticality as revealed by avalanche analysis, but you cannot trust exponents. It's very odd state of affairs, in my opinion. It's very, so I was, at the same time, I like to tell this to Taiwan, very pissed at him and very proud of him, of course. So he challenged our results, and he was right. That's nice. I don't know if I should give this as general advice. So challenge your advisor whenever you can. Not whenever you can, but I mean, it's a healthy thing to do, right? Yeah?
It's certainly true that of the things you showed us, some of the power laws were better power laws than others.
Yeah.
Is there any sense that when you get to the crossing point, it's a better is there anything systematic going on here?
Yeah. So the power laws here are the best. Here in this low CV range, they're debatable at best. So they still pass some statistical tests, but so if you believe the statistical tests, they are strictly power law of some exponential decay, but no, they're better here. Yes. So there's that. And again, you get results which are compatible with each other within error bars. So the subsample model of a beam field-derived percolation model explains the data. So this mean field-derived percolation model, subject to subsampling plus CV parsing plus binning, can lead to apparent exponents which differ from the true ones and reproduce results for anesthetized rats. Despite the distortion exponents, the cracking noise scaling relation still satisfy to my despair and our despair. Come on. Maybe it's obvious, I don't know. But this is true only if the model is within 2 to 3% of its critical point. You cannot go much further away. So there's that. So I insist, and I keep rediscovering that. I had the same problem in 2010 as older paper subsampling, and subsampling comes to haunt me again. There's this methodological vulnerability. So most importantly, for the rest of my lecture today and my lecture tomorrow, is the fact that this narrow interval of the control parameter yields a wide range of CV, of population firing rate, and therefore CV is a poor control parameter. What I mean is that these different colors here are tiny variations of the G control parameter of the model, and this gives rise to this wide spread of CV values. And that, the fact that CV is a poor control parameter, I'm being very generous. It's not a control parameter. It's a lousy control parameter. It's not at all a control parameter, which is something we had in the back of our hands as interpreting the experimental results that I showed you before. This completely reshapes how we look at the results. So let me go back and revisit the plots that I showed you before. So if you're 3% off criticality, results don't match, the model doesn't explain the results any longer. In order to do this, so you're varying your G values very close to the critical point, and this very tiny variation in control parameter gives rise to this wide distribution of CV values. Okay? This is what the model does. So a very simple model, by the way, without adaptation, so the model is static, right, in its parameters. So if you go back to the experimental results, and remember what I said when I mentioned this residence time. So according to the scaling relation being satisfied, this have the gray region where the system is critical, or that's how we interpreted it back in 2019. So this residence time tells you that the system is a criticality en passant. So but when this paper was published, many people asked, so what you're saying is that the system is critical, but most of the time it's not critical because it's here, it's here, it's here. I mean, the most probable one is here, but it's passing. So what about your reasoning, criticality reasoning, rationale is critical all the time, whatever. Actually, now we know, I mean, if the model is worth anything, I don't know if it is, but if judging from the model, that's why simple models can be interesting. This whole thing here actually corresponds to very tiny fluctuations around the true critical point, because this I know, I have calculated. This is an analytical model. So the interpretation according to the model is not that you are at criticality just in passing here eventually, but actually you are all the time near the critical point. So it's a different interpretation. Right? Because you are all the time here. If you're not here, close to this guy, this thing breaks down. So I like this one better. I mean, if you're into criticality, oh, this is a signature that the guy is close to this critical point. But then, and then there's a corollary, second-order consequences of this interpretation here. If you are all the time near a critical point, these so-called synchronous states and asynchronous states are fluctuations around the critical point. This opens a can of worms in the discussion with, with people in the literature, because that's not something they would agree easily. I'm saying that I can explain both very different kinds of regimes with one mechanistic, quote-unquote, explanation, that being around the critical point gives you, well, we know that at a critical point you have these large fluctuations, and what I'm seeing in this model is that once you subsample, what you're going to see is that sometimes it will look very synchronized, sometimes it will look very desynchronized, but just by chance as fluctuations of this critical point. Anyway, of course, here I'm talking about experimental results, criticality en passant. These are experimental results, which as such have the values that they have because the experimental results. And this is just a model for whatever value you attach to it, right? And so what I'm going to do in full tomorrow, well, can I test this idea of all the time near criticality experimentally with a different technique than avalanches, because I know the problems now. So I can compare bananas with bananas. I mean, experimental results, just a different technique. So tomorrow I'm going to tell you what happens if subjective data to the renormalization group that Bill described yesterday, which people came to call phenomenological renormalization group. I don't know if you call it like this, but people do. So to have a clear picture of what's going on, okay? So this I'm going to do tomorrow. Today, in the last 10 minutes that I have, just going to revisit the maximum entropy that Bill mentioned also yesterday. So you have some entropy that you want to maximize respecting some constraints, and then you calculate the specific heat to see whether it has a peak. Remember, he showed you this yesterday? And except here we're going to use, since we have this data which has oscillations and whatnot, this thing goes up and down, we are using this kind of energy in which have the average, the global firing rate compared in different timestamps. This seemed more natural to us to do, okay? So very quickly, what I'm going to show you is that we take the same data and you calculate the co-occurrences of those things, and this thing, you maximize entropy respecting the experimental constraints, like Bill explained brilliantly yesterday. I won't go through the steps again. And you obtain this probability distribution. And once you do, you can calculate the specific heat, and then you introduce an artificial beta function here. So the data is described, the probability distribution describes data for beta equals 1. And you change beta, have a family, an artificial family of models, and you can check whether at beta equals 1, you are at the peak of the specific heat, which will be a signal for criticality, depending on how this peak scales with system size. Right? So I'm looking at the same data as before. Now, let me start with the whole data. Forget about the CV stuff for a while. The specific heat is a function of temperature. This temperature T equals 1. As you increase the number of neurons, you have a peak that keeps growing and growing toward T equals 1. So as far as maximum entropy signature of criticality go, this is a very nice signature. You can do rat by rat here. This T star will be like the peak of the distribution. They all go to 0. They all go to 1. And importantly, I don't remember if Bill showed one of these plots yesterday, but if you take the data and shuffle your data, everything, so you destroy the correlation and stuff like this, you don't get a peak at 1, okay? It's not that any random bunch of spikes will give you that peak. The real data does. But if you shuffle the spikes and therefore destroy pairwise correlations, then you don't get this. You get this kind of ugly thing here. Okay? But that's not what we did with the avalanches, is it? What we did with the avalanches is we parsed the data by CV. So let's redo maximum entropy, taking slices of data in which CV has some interval value, and redoing everything, and doing whether this scenario of the peak specific heat corresponds to, gives a signature of criticality or not. So if I parse by CV, then I have a family of specific heat curves for each value of CV. So you see here that for low CV, 0.55 or so, the peak is off T equals 1. I increase CV a little bit, the peak increases. I increase CV even more, the peak increases, close to 1 here. If I keep increasing even more, it goes away from T equals 1. So you can make a plot of the peak temperature here as a function of CV for different rats, and it's close to 1. It's not too far away, but it's closer to 1 here. And then it goes above. Whether this is all critical or not, it's hard to say. You can define, you can be, well, is this small, is this far away from 1 or not? Well, as compared to what? So we try to something like some dimensionless distance to criticality, like taking this, let delta T be the half height width of this distribution, this CV curve. And so when we compare the distance to 1, we normalize it by the width of distribution, so that comparison kind of is measured against something, right? Let me try to make a drawing. If I have 2 peaks, one is like this, and the other one is like, so this is a peak, and this is a peak. Peak number 1, peak number 2. Which one do you think is closer to? Which peak is more relevant, right? To this. This one is closer, but it's much wider. So we try to just have a measure of how close they are. So if you choose this kind of normalized distance, it just changes the data a little bit and make this data more or less collapse into this curve. And again, what is interesting here for discussion, to make things more confusing, perhaps the fact that if you look at this CV range in which the peaks are converging to 1, it is precisely, well, it's not precisely. It has this value with some standard deviation, which is then close, borderline compatible with this one, which obtains via avalanches. We had these problems. So perhaps CV is creating more problems than this parsing by CV is creating more problems than it is solving. But in our defense, the reason we did that in the first place was because, again, we were informed by the language in which the neuroscientists are doing their experiments. So they talk about this thing. They segregate synchronous from asynchronous behavior. They are interested in these things. They regard them as different states. They call states. So we thought it would be wise to do this. But apparently it is creating more problems than it is solving. So here you see that we have the results here for maximum entropy, suggesting that yes, it's more or less the same spirit of this thing here. Yeah, but we also have this for the whole data, right? So this thing suggests that it is critical all the time, right? That's how I interpret this plot. Whether this one, if I artificially parse, well, suggests no, only when you're passing through this. So maximum entropy is not helping us much here, but it's a nice result. So we love to test it on our data. This is published to 2020. I think it's not there. Sorry, I forgot. Anyway, so I hope I have convinced you that neuronal avalanches, especially for spikes, are methodological problematic. So we're going back and forth, giving nice results, which then turn out to be not so nice, etc. Tomorrow, I would like to revisit the contrast between this that we get, this message we get from avalanches, this message we get from the model, by using the phenomenological normalization group. Okay? Thank you.
Hello, Professor. Thank you for the lecture. I find it very interesting to see all those results in scientific work. So I have a question about the. Let me search here about the scaling relation of the critical exponents.
Mm-hmm.
Where does it come from? It comes from the theory, of course, but why is it that way?
Maybe I should have studied the deduction of the scaling relation to show here. I'm not sure I can do it like this. Where is it? This one, you mean? Yeah? Yeah, the one is written on Blackboard, right? Essentially, I'm not going to try the deduction now, but if you have a distribution like this, I'm sorry, it's easier here. You integrate, say, you have distribution like this. So if you integrate from some size s to infinity, you have all the fraction of all the avalanches that is larger than a given size. If you integrate this one, you have all the avalanches which are larger than some which have some lifetime longer than some minimum, the thing you're integrating from. So but you counted the number of avalanches in both cases, so it should be the same. So we will integrate those two things, it will give you minus tau plus 1, minus tau t plus 1 as the result of integration. And then you assume that the probability of an avalanche having a given size, a certain size, given some duration tau, then this is not a power law. It's supposed to be more or less peaked around the mean value S given tau, because if you have a very long avalanche, it cannot be too small. It just can't. And once you assume this is peaked, you make an approximation and you get the result. I mean, I'm just sketching but it's an easy calculation. Just don't want to do this here and It's being recorded. I could make something. I don't know. But, so it's just, if you take a course on phase transitions, this is one of the first things, well, not the first thing, but you're going to do eventually at some point is just playing with those scaling relations that I expect here and there, and well, they are connected. And then the exponents start talking to each other. This is one scaling relation, but there are many scaling relations. And I consider that to be I marvel at the intellectual beauty of these things. So before the renormalization group, of course, which is like crowns everything, you have these calculations, which are very, if you think about it, you're doing the calculations, you're very far away from the phenomenon, the original phenomenon now, and you're playing with mathematics, and it works. I mean, so I find it beautiful. I don't know. But again, statistical mechanics, I don't know, it's beautiful. I don't know if I answered the question.
Oh, yeah. I have one more question, actually. Don't know if it's quick or no, that depends. But, well, after hearing your lecture, I realized I had an understanding issue with some results from the last lecture, actually. And while you were talking about the last lecture on your first slide or something, you showed that the freely behaving rat, and you do all the data analysis and such, it does not, the probability does not obey a power law. It obeys a log normal law, and if you anesthetize the rat, it kind of obeys the power law. That's it, right? But and if you take the model and you undersample the model, it kind of goes to a log normal, yeah?
Yes.
So that means that if undersampling occurs, either the rat is anesthetized or not, right? If it's freely or anesthetized, undersampling,
It's always there.
it's always there.
Yes.
So either the undersampling works only if the undersampling disturbs the distribution, only if the rat is freely behaving.
Yes.
Or
But not for anesthesia.
but no, when it's anesthetized.
Not for ketamine, xylazine anesthesia, which were the anesthesia of yesterday. Not urethane. So yeah, didn't specify. Yesterday I was talking about ketamine, xylazine anesthesia, which in very strong doses, by the way, so generates the very synchronous activity that I showed you yesterday. Urethane is different, because urethane gives you precise. That's why some biologists like it, because it gives you kind of everything, a little bit of more synchronous and asynchronous behavior. But I'm sorry, I interrupted you. Yes.
No, I just wanted to know if the undersampling, it doesn't work on the ketamine anesthetized rat, or it kind of works to bring the rat to the power law instead of from
That's
the
an excellent question. That's an excellent question. Once you use undersampling to try to save face and explain the log normals that you see in free behaving animals, you create yourself another problem. Well, yeah, why? Why you don't get the distortion in the anesthesia? We suspect it has to do something with this oscillation that you have in anesthesia. Because oscillations, this synchronous behavior, in a way, what they do. Oh, if you're measuring somewhere, you're measuring everywhere because they're kind of synchronous. So that would perhaps attenuate the effects of undersampling. And in that, I didn't show the results yesterday, but in that paper, we try to mimic. It's a very disparate effort. So we try to take one of these direct propagation models at criticality, and we put some oscillating fields to kind of make this thing oscillate artificially. And then you measure the avalanche, and then this subsampling effect is attenuated, so you get more or less power. But it's a very ugly solution. I mean, something that doesn't oscillate, you make it oscillate by hand, essentially. There are more interesting solutions, like the cross model that I showed you yesterday, which kind of oscillates on its own, or we saw here that a mean field repopulation model under subsampling will not oscillate, but will show synchronous activity. So in physics, sometimes synchronous activity and oscillations are confounded, but they're not the same. One thing is synchronous, I mean, go together, but not regularly. If you have an oscillation, you have some kind of regular behavior, which is also synchronous. So I don't know. Did I answer your question?
Yeah.
No?
It's a good discussion. Yeah.
Yeah.
Doesn't actually have an definite answer, I guess.
Yeah, the thing is that I don't know the answer to most of your questions. I mean, I don't. We don't know why subsampling does not affect the ketamine xylazine power laws like behavior, but we guess it has to do with that.
Thank you for the nice talk. I wanted to ask you about the end of the talk with the specific heat and all of that, because I got a little confused when you said that you analyzed all the data and got those beautiful curves, that specific heat versus T. So that's from data, those
from data.
very nice curves. Okay, so from there you sort of deduce that you are near criticality all the time. Is that right? That's what you argued somehow. But then you went to analyzing.
Hold on.
So maybe I am confused.
Yeah, me too. Yeah.
No, because
then
so when you,
so this is one possible.
Let me tell you what I wanted to ask. So this seemed to favor your idea that you are near criticality all the time, and that by either undersampling the data but also parsing with these coefficient of variations, you got results that didn't agree with this idea that you're near criticality all the time. So it seemed like this analysis was supporting your model, but then you didn't conclude that. So I got a little confused with that.
We were also confused. Actually, to make things a little bit worse, this paper came before the modeling paper. This one came before. So we had no idea that this interpretation would then be attacked by ourselves, I mean, with a model that challenges the discussion, right? So that's why I hesitated into bringing these results here today, because I know they're confusing. Your question is very accurate. It's very confusing. And we didn't discuss the paper because we didn't have an issue. It hadn't come up yet.
I mean, reassess your conclusions here, like saying, well, in view of the model, then I can say that this is because we are tweaking the data in a way that affects these results. Isn't this supporting your model?
Perhaps it is, yeah. So I tend to have this, look, it is not in the Avalanche paper, but I asked Antonio Fontanelli, the first author, specifically, if you take, the whole data and do avalanche analysis, do you get nice power laws? And he said, no. That's why you don't see the result anywhere. But I tell you, it should be in your PhD thesis. That's where you put, that's a kitchen sink when you put negative results. But they're important. So no, right? And which to me just reinforces the methodological vulnerability of avalanche analysis, for spikes at least. It's just a mess. Here, you can have two interpretations, I think. Well, just take the data, forget about CV, don't care. Again, I insist, we did the CV parsing because we were informed by the literature in junior science that this is an important thing. Maybe there's a transition there, right? Between synchronous and asynchronous. Is there a phase transition in between? I don't know. Right? I mean, it was a legitimate question. There was legitimate reasons for doing that parsing. If you don't do anything, just do maximum entropy. Well, yes, I think it's a nice signature to criticality. You think, Bill, is a nice signature? probably 20 milliseconds or 30 or 10 or 50, I forgot. Maximum 50 milliseconds, perhaps. So maybe this is just a subtlety and which is introduced by artificially parsing this thing. And if you want to stick to the view that is critical all the time, then you interpret this thing as being too small. Actually, this is essentially one. Close enough to one. Or maybe you need to have more neurons, then the peaks will be even closer. So it will be something like this. I think here we are working only with the maximum number of neurons, which is like 50 or so, I don't know, that you can do to run the thing. And so thank you for your confusion. I share it with you. And what I like best is the results I'm going to show tomorrow with the renormalization group. And I leave it as a kind of primer for tomorrow, the fact that he mentioned the log normals of yesterday. And you could argue that, hold on. That these are log normals too, right? In a way, this is very low CV. It's probably well fit by log normals. So, in both data from yesterday and data today, these very low CV values then fail to give signals of criticality. Okay, this very asynchronous behavior for ketamine, xylazine anesthesia, and for urethane anesthesia. When you have very synchronous behavior, the signals for criticality go away. And even for maximum entropy, this is when they are the worst, right? So here, if you insist on parsing by CV and look here, then yeah, they're low CV, they are the furthest from the guy. I insist on that because tomorrow, I revisit first this data, and with the phenomenological renormalization group, and I will go to free behavior data with huge number of neurons, with neuropixels. And animals, when they are awake, I just showed from the review, they're asynchronous all the time. So according to these previous results, our intuition will tell us, yeah, nothing is critical here. And spoiler, also everything is critical there, apparently, if you do a different analysis technique. So there are differences between asynchronous activity under anesthesia and in the freely behaving animals that you cannot see with your eyes, but some techniques can tell apart, very interestingly. So this is for tomorrow, but you had a question, right?
If you took a statistical mechanics problem that you understand, and you do Monte Carlo, you get lots of samples, and you compute something like the specific heat, and you see the peak as a function of temperature, and the peak is in the right place. There's a finite size correction, of course. But suppose that you decided to take your Monte Carlo samples and parse them in some way. You can't quite do CV for a ferromagnet because the magnetization's 0 at the critical point. But, I mean, for example, if you parse them by the variance of the magnetization, that is actually the susceptibility. And so in some funny way, you're choosing a subset of the data which can't be critical, right, if you pull away from them. So I'm wondering if there's an analog, if you could sort of go down that path of take a system which is genuinely critical and then do this parsing and show that the thing which is supposed to peak at the critical point peaks away from where the system, as a function of the control parameter. You understand what I'm saying?
Yeah.
I mean, it seems like you've almost done that.
Yeah, well, like I said, this paper came before the subsampling paper, theoretical paper, model paper afterwards. And we didn't go back and check, but we could just do the same, right? Just repeat exactly what we did with avalanche analysis, parsing by CV, with this thing, and to see what happens. This could be done. It's a nice idea. It's just that then your papers came out with this phenomenological renovation group. We were very excited, and it kind of works very well. We just abandoned the idea, but maybe we could go back. Yes.
All right. So I think unless someone has a very short question, for the sake of sorry, for the sake of time, we can stop here. Thank you, Mauro, again for the lecture.
Thank you.